Everybody, can we get everybody's attention for the next session, please? We're going to start shortly. Okay, so hi everyone, welcome back. This next session is lightning talks. So these are talks, short talks that are selected from many of the interesting abstracts that were submitted for the meeting. And we're going to have each talk be around 7 minutes with maybe one question afterwards. And then we can, so speakers, you can connect with the speakers at the breaks for more questions. So I'm going to start by introducing Mijo Simunovic.
Very good.
Okay, who is from Columbia University. So go ahead, Mijo.
Hello. Hello, everyone. Is my mic working, or am I just really loud? But I'm really loud. How about now? Yes. Wonderful. All right, let's begin. Okay, I am a professor at Columbia University. I am kind of at the intersection of physics and developmental biologists, and we're really opening up to the fetal atlas community. I really hope to also meet a lot of you here, and hopefully some of our work will also stimulate potentially some of this. So let's just dive right into it. And what we're really interested in is understanding mammalian embryonic design, right? How do we go from kind of using all the same signaling pathways and same morphogenetic pathways to somehow end up as completely differently looking organisms, right? An elephant has never kind of become a mouse. There's never a human embryo that looks like a cat embryo. That's never really happened, right? So how come? And really what's unique about mammals and what is also bottleneck in mammalian pregnancy is implantation. So we really believe that a lot of these symmetry breaking processes occur right after implantation. Now this is also the so-called black box in mammalian developmental biology because we don't really have access to observing mammalian embryos after implantation. It is also a major health crisis, so there is certainly a pragmatic reason to study it. It fails 50 to 70% of the time of human pregnancies. It is also very much the cause to the vast majority of pregnancy, not just pregnancy failure, but also pregnancy disorders like the placenta accreta spectrum disorder we heard about earlier today, or the ectopic pregnancy and other issues. Of course, there is a huge gap in knowledge. One is a technological gap. We simply don't have access to mammalian embryos, but also it really needs to be mentioned that there is also very poor interest historically in funding women's and LGBT health in general. Okay. So why study human post-implantation development? Or how can we study human post-implantation development? One, we can use human embryos, and this is something we've been doing for a while in the field, but it's really technically and ethically challenging, of course. So why not just create models of the human embryo, right? And so this is precisely what we do in the lab. We use human embryonic stem cells, and we try to create components of the human embryo and just put them all together. So this is a component-based sort of approach, an assembloid approach, if I may, in which we essentially create individual components of the human blastocyst or the human post-implantation embryo, try to put them together. So I'll give you an extremely brief overview of about 7 years of work, which starts with the first component, which is making the human epiblast, so the intraembryonic tissue, and we found a combination of PEG-based hydrogels, these wonderful chemically defined hydrogels, and three-dimensional tissue culture environment where we're able to grow human embryonic stem cells into three-dimensional structures that self-organize into something that really resembles a 10-day-old post-implantation human epiblast. So on the right, you see a comparison between the actual in vitro cultured human embryo epiblast and the human model of the human epiblast. Okay, so now that we established component number one, what about the really important component, and that's the extraembryonic tissue, right? So at this stage of development, human extraembryonic tissue is composed of two key components, the hypoblast and the trophoblast, right? So over the years, for a long time, it was really difficult to get these components because there was simply no way to essentially differentiate stem cells into them. And so luckily, over the past years, new ways to differentiate stem cells, human stem cells into extraembryonic cells have now emerged. And so we're using these kinds approaches, and we've modified some of these approaches to essentially differentiate human embryonic stem cells into both of these cell types, the trophoblast and the hypoblast. And this is just for you to show that we are indeed able to get cells that resemble the trophoblast as with interspersed hypoblast cells. And now, by using these cells and combining them with a human epiblast component in sort of highly engineered way, such that we can control the titration and stoichiometry of these tissues, we're actually able to get really highly reproducible three-dimensional structures that resemble the early post-implantation human embryo. If you look a little bit closer and compare it to the actual 10-day-old human embryo cultured in vitro, you'll see some important similarities, right? So it has this epiblast, right? This green tissue is the cavitated epiblast tissue. It's surrounded by layers of hypoblast and trophoblast tissues. And furthermore, it seems to recapitulate even the three-dimensional morphogenesis of these tissues, and that's really important. So even not just the gene expression and the correct tissue juxtaposition, but also the morphology of these tissues is reproduced. So what happens in 3 more days, or 1 to 3 more days? Now, we don't provide it with any exogenous signals, yet this model spontaneously is now able to break symmetry, right? This is what happens normally in development. After a few days after implantation, the epiblast starts to form the primitive streak asymmetrically on one side of the embryo, right? And this is precisely what we're able to recapitulate. And even if we leave it for a few more days, we can even see that not only did we break symmetry such that see this green tissue on one side, the brachyuria expressing tissue, the future mesoderm, is also actually able to reform new layers of epithelial tissue, right? And on the other side, we have SOX2 positive tissue, or the sort of future ectoderm tissue, which also started to form various interesting cavitated morphologies. If we, of course, do our single cell analysis here, we'll see that we indeed recapitulate some of the most important tissues of the post-implantation human embryo, and certainly we've been able to compare this tissue with the currently one existing data set of a human gastrula, and there is really, really wonderful overlap. Okay, to kind of zoom right through a lot of this work, I'll just say, what are the kind of next steps in this? Sure, we are now creating we are modeling human post-implantation development, but there's of course a limitation to how long and can these kind of structures even survive outside the womb, essentially. There certainly is a biological limitation to that as well. And we truly believe that there is, and based on evidence largely, that there is probably a communication between the endometrium and the embryo. So what's the solution? Well, let's create the endometrium, right? So certainly there's now a way from Turco Lab and others to actually create endometrial organoids. So this is certainly of interest, but actually we started this research at the beginning of the pandemic, and we didn't have access to human subjects for a long time because we were not allowed to. And so we decided, okay, how do we overcome this issue? So let's just try to figure out a way to create endometrial organoids from embryonic stem cells, right? Actually, no one's ever done that before, right? You have all the organs, probably most of the organs had now been recapitulated from ESCs except for the reproductive system. Again, not a big surprise, right? So what we did is, knowing the development somewhat of the female reproductive system that starts essentially through mesoderm and eventually diverges into the kidney, we figured out a way to block kidney development and essentially create the Mullerian development, which eventually gives rise to the oviducts, the endometrium, the vagina, and the cervix, right? And so this is what we did. And we actually, not only that we were able to get endometrial epithelial cells, but also endometrial stroma from embryonic stem cells. Once recombined, they actually recreate the endometrial kind of niche, which we're now able to co-culture with our models of the human embryo. Okay. One more thing, just to kind of give you an overview where this is going. Essentially, now we have access to not only study early post-implantation development, but also to really engineer more sophisticated microfluidics approaches where we can even deliver signaling gradients such that we really recreate this kind of proximal distal axis in endometrium and essentially study this two-way communication between the embryo and the endometrium. Yeah, and with this, I'll just say that we can potentially even combine this with high-throughput CRISPR screens and essentially study not only what cell types are present and when they merge, but also how they interact with one another and what is the hierarchy in signaling that drives cell type formation. In other words, molecular mechanisms, as we like to say. And with this, I'll just end, and I'll show you my wonderful lab, and who is responsible for this research. And yeah, I'll thank you all for your attention.
Yeah, wonderful talk. I was very intrigued by your endometrium studies. To differentiate it into endometrial cell types, do you use anti-Mullerian hormone? This is my first question. So do you know, anti-Mullerian hormone is actually the hormone that is necessary for the
You don't want to use the anti-Mullerian hormone, right? So anti-Mullerian hormone is going to repress Mullerian development. It's secreted by the SR, by the Y. It's Y encoded, right? So every individual is both male and female reproductively at some point until the anti-Mullerian hormone kicks in and shuts down Mullerian development, right?
It shuts down.
So have we used it to verify that we have Mullerian tissues, for instance?
Exactly.
Yes, we have. It was fun. Yes,
Okay.
It was a fun experiment to do.
Yeah, that was my first question. And the second one, in your culture, in your model of endometrium, I haven't seen any smooth muscle cells. And
we don't get myometrium, is what you're asking.
Okay, and also glands.
Glands we have.
You have glands,
yes.
But no muscle cells.
No muscle cells, they're myometrium, right? So we don't, for now, see myometrium. Probably we would have to, I don't know, maybe enhance and not sure what we'd have to do actually.
Okay, thank you.
So there's one more, there's a virtual question, so we're just going to take the virtual question, sorry, because it's the first one that we've had and we want to encourage participation.
Yeah.
We'll have to move on, sorry.
How did you block the kidney development?
Trade secret? Can I answer this way? It's FGF signaling.
Okay, sorry, I think we have to move on. I apologize. So please find the speakers in the break session. Okay, so thank you very much. Okay, our next lightning talk speaker is Jonas Fleck from ETH Zurich.
Cool.
Does this work?
Yeah, sounds like it, right? Okay, cool. I'm Jonas. Until quite recently, I was a PhD student in Barbara Treutleins lab at ETH. And now actually, as of 2 weeks ago, I started my postdoc at ITB at Roche. And today, I will talk about my PhD work, which was on perturbing and inferring cell-fate regulomes in human brain organoids. So I think a driving question, especially at a meeting like this, is how do you get from one stem cell to kind of these complex organs like the brain with these intricate structures and like kind of this vast diversity of different cell types. What kind of magic happens in between? What is the gene regulation that's required to make this happen? Also this seems to be kind of commonality here. We've chosen organoids to model this and to research this. Organoids are really useful because on the one hand side, we can grow them in the lab from stem cells. We can manipulate them. We can perturb them. And also, they recapitulate a lot of the features of human brain development that we really care about. One is, for instance, like this morphology. When you look at these beautiful ventricles with progenitors and neurons that you also see during human brain development. But also, if we do single-cell RNA-seq on this, as we and others have done in the past, we can see these regional trajectories, right? There are these neurons that look like cortical excitatory neurons, and others look like diencephalon, midbrain, or hindbrain neurons. And so the driving question in my PhD was really, how do we get there? How do we get from this ball of stem cells to this heterogeneous regionalized tissue? And specifically, we were interested in the gene regulation behind this. So for this, I teamed up with a fellow PhD student in the lab, Sophie, and she really spearheaded all of the experimental work behind this. And we did what people usually do here. We made an atlas. Specifically, we made an atlas, a multi-omic atlas of organoid development, starting at pluripotency up until day 30 and day 60 old organoids when we have these regionalized neurons. At each time point, we'd sample it quite densely. We took RNA-seq, we performed RNA-seq and ATAC-seq from the same suspension to really get into the gene regulation there. And then we could integrate these two modalities into meta cells that essentially then have kind of these summarized cells that have both modalities. If we lay this out in a UMAP, we see this trajectory from embryoid bodies over neural progenitors, which then branch out into these three neuronal clusters that you can see on the right. And these are really these regionalized neurons that I showed you before. So now we wanted to resolve the trajectories that lead up to these regionalized neurons. So here we used two really useful tools. One is RNA velocity, and in conjunction with cell rank, which Dana already introduced earlier really nicely. And we used this to get transition probabilities into each of these terminal clusters. From these transition probabilities, we could then construct this graph of high-resolution clusters, and this then really nicely reveals this hierarchy, where first from the neuroectoderm, we branch out into non-telencephalon and telencephalon, and this then further diversifies into dorsal and ventral components. And this is really similar to what we see during human brain development, right? So now we wanted to also see what does the neuroectoderm look like when it first diversifies here. So for this, we actually gathered data from additional stem cell lines and also performed multi-ohm sequencing on this. And one of the first things we can see when looking at this data and comparing it to references is that there's a clear anterior-posterior axis in there. Some cells become more similar to forebrain, others become more similar to mid and hindbrain structures. And we can also look at the expression of transcription factors and ligands and receptors that are involved in patterning in the brain. And we can see that the expression in some of these populations are actually really similar to expression patterns in mouse organizer populations. And not only that, we can also see that these expression patterns, while the proportions of different cell lines in these clusters differ, we see that actually the expression patterns are quite reproducible between stem cell lines. Because this is multi-ohm, we could also identify these cis-regulatory elements for these genes and see that also these follow kind of a quite striking pattern. And interestingly, these were more variable between stem cell lines, so this might also have some implications on why different stem cell lines preferentially make different regions. So now having this gene expression and accessibility data together, we also wanted to go into the inference of regulatory networks. For this, we developed an algorithm, a tool called Pando. And essentially, it works by building models of the expression of a target gene by considering the interaction between the transcription factor expression and the accessibility of its binding site. And with this, we really leverage this relationship between accessibility and expression. Using this on our data set, we can infer this GRN of organoid development. And I'm not going to go into too much depth here. I can explain this maybe later more. But we can now use this GRN also to go into these really regionalized sub-GRNs that are constrained by accessibility. Now, our next question was which transcription factors are really responsible for setting up these regionalized trajectories. For this, we performed a pooled CRISPR screen known as PROP-seq or Perturb-seq. And we perturbed 20 transcription factors that are known to be involved in cortex development. And we found that actually most of these transcription factors had a significant impact on the regional composition of these organoids. Specifically, I want to highlight CLE3 that really had kind of a striking depletion in the cortex. And we also validated this in CLE3 knockout lines where we basically didn't see any dorsal telencephalon at all. So now we were wondering if we could combine, really draw the GRN that we inferred earlier to get into the gene regulation that happens there. So first, we used our CRISPR knockout lines to perform multi-ohm sequencing on this neuroepithelial stage. And you can see in the UMAP, this is right the time point where we see non-telencephalon and telencephalon, but the telencephalon is not yet dorsalized or ventralized. And we also see that the strongest dysregulation of CLE3 knockout happens in exactly these telencephalic progenitors. So these are likely the changes that later lead to the deletion of cortex. And now we can use these differentially expressed genes in conjunction with the GRN to find the subnetwork that is, from the direction, completely consistent with the DE that we see. And that really gives us this hypothesis, this model of how CLE3 regulates cortex development. Specifically, we found two novel downstream targets of Cli3, Has4 and Has5, that we also validated using cut and tag. So in summary, I hope this was kind of interesting to see that these atlases, especially multi-omic atlases, can really be used to disentangle gene regulation that really underlies development. And with this, I want to thank everyone who was involved in this project, specifically Sophie and Chisong, but of course also Barbara and Gray and everyone in the lab. They're amazing people.
Thank you. Great talk. Any questions for Jonas? Okay. Yeah, fine. Okay.
Hi, thank you very much. That was really interesting. I have maybe a technical question, but basically, why did you feel the need to develop this algorithm? Were there no other alternatives? And have you tried comparing it with Scenic Plus?
Actually, where's this question coming from? I don't know it. But yeah, so Scenic Plus wasn't really developed when we developed this algorithm. I think Scenic Plus really tries to achieve something quite similar to this. I think at the time of development of this, there wasn't really an algorithm out there that could really leverage single-cell data that was paired, that both had a tag-seq and RNA-seq data. Most existing such algorithms were only based on RNA-seq data. So we thought kind of this new algorithm could really leverage this relationship between the two modalities when they're measured jointly. Yeah.
So maybe one quick question from online and then do one more question.
Thanks. How do you make sure the TF is in the active form in the Pando pipeline? Existence of the TF in open chromatin doesn't mean transcription initiation.
Yeah, that's right, and that's kind of quite a big limitation of a lot of these GRN inference mechanism or GRN inference algorithms is that actually we don't know a lot about kind of how the transcription factors really, and that also goes into what was discussed earlier, that we don't know also how transcription factors interact between each other, and that could really change how they regulate. So really more information there would be really beneficial, but currently we can't really take this into account.
Okay, I think we'll move on to the next speaker, and thank you again to Jonas. Jonas. So our next lightning talk speaker is Paulo Czarnewski from the Karolinska Institute.
Yeah. Can you all hear me? Yes. So hi, my name is Paulo Czarnewski, and today I'll present a little bit on the efforts that we are trying to establish for the first trimester Human Developmental Cell Atlas. So I'm also part of the National Bioinformatics Infrastructures. I will come to that in a second. But today I'm using my hat as scientific coordinator for the Human Developmental Cell Atlas in Sweden to present a little bit of our efforts. So this is just a brief overview of the cohort that we have. We have many different technologies, and we're collecting embryos at all the different time points as possible. So here you can see a map of all the different samples and how many samples are collected for all the different omics. So ranging first from like week 4 until week 13 and 14 maybe. So most of the results that I'm presenting, or at least mentioning now, are result of this big cohort. So we're focusing mostly on the heart, lung, spinal cord, and the brain, and there are different groups involved in that. So to start with spinal cord, so we also collected spinal cord from different regions and a different developmental time. So performing not only single cell, but also spatial transcriptomics and in situ sequencing for cellular resolution in space where those cells are, and also mapping that across all the different time points. So you see like from week 5 to week 12. There is also a very intricate spatial patterning of many different cell types, so cholinergic neurons, inhibitory neurons, excitatory neurons. And we have also a preprint by Xiaofei Li And he's actually here today, and he's presenting a poster session number 2, poster 16. And if you want to get more details on that, please go check him out. So one of the other highlights in this paper is also combining not only the data produced in-house, but also combining many different available data sets, totalizing around 2 million cells, and then doing the integrative analysis and see how these cells differentiate over not only the first trimester, but also the second and the third one. So for instance, we detected oligodendrocytes, which are, and the progenitors of these cells that are already present in the first trimester, which was not previously known. A second story is about the lung. So this is also a very comprehensive mapping of lung at all these different time points, not only doing a single cell, but also spatial transcriptomics. And the most important thing here is that we actually have the right lung that was processed for single cell, and then the left lung that was sequenced for spatial transcriptomics. So we have paired samples of single cell and spatial transcriptomics data, as well as in situ sequencing screenshot analysis. And in there, they identified several molecular mechanisms and intracellular communication patterns, mostly FGF and Wnt signaling, or as SOX2, SOX9, that defines the niche and differentiation of the branching of the lung. Many of those results are described also in the bioRxiv paper. And if you want to get to know more about that, Alexandros Santolis and Sergio Salas is also here, and they will be presenting poster 17. I think this session is tomorrow, probably. Additionally, we are also trying to build the heart version 2. So in the beginning, we had just a very brief overview of the different developmental stages of the heart. It's still only 3 weeks, but now we have many more samples collected, many different cell types also captured, because you have depth into capturing these rare cell types. And we actually can find even the spatial patterning of these different cell types at the different parts of the heart, even tracking the conductive system. And if you're more interested in that, so Shaneta and Julia Freuer, they are also here today. So they have also another poster session showing some highlights of these papers. Another work that is also in a bioRxiv is also from Sten Linnarsson's lab. He's not presenting this today, but if you have any questions, ask him. That you have a complete characterization of the developing human brain at different parts. So the intricate part of this project is that the brain was already desiccated and collected at individual regions, and then processed for single-cell RNA sequencing, so then you know exactly where the cell is coming from. And then based on that, you can identify which signals are the ones that drive differentiation of neurons at the different regions of the brain. So we have midbrain, hindbrain, the thalamus, and so on. And then you can also map the different cells across the different regions of the embryonic. And here he used IL, which is another technique developed by him to profile single mRNA molecules in space. And just to briefly mention, this is another work that is not presented here today, but is also an ongoing effort in our institution, is to create this common coordinate framework and references, and how to map multiple samples to the common reference. And that is done by Gaussian process, which is a very flexible approach to really combine different tissues in different orientations, or like lacking parts of the tissue, so that we have basically a computational framework to project gene expression into H&E images or into these references. In that case, you can see on the bottom there, the composite part, different genes being expressed, but that reference is actually just a outline of how it should be, how the tissue should be, rather than a physical tissue. So just to demonstrate that this model can actually even predict gene expression to whatever image that you would like that kind of makes sense. So that makes it way easier to interpret how you evaluate gene expression, so you can clearly see how they are distributed rather than looking at many different tissue slides. And that's also a paper in bioRxiv already there. Another tool that we are very proud to be collaborating with is with tissue maps. So tissue maps is a tool developed by Karolina Welby in Uppsala University, and we use that for visualizing our data sets. So we can, as of now, handle around 10 million points, very large images, those that cannot even open in QPath, they can already be done there. And it runs on any different technologies, so if you just have points into the space, you can basically import that into tissue maps. So Christophe Avenel is also here presenting a poster session 2 as well, and poster number 2. And basically, this is just one of the overview of the UMAP that we have. So for example, in the top, you can see single cell, spatial transcriptomics, in situ sequencing, and many others. So you can see that basically we can plot everything using that. Besides producing data and publishing papers, we also are highly engaged in training. So this year in May, we have this EMBO course to teach spatial transcriptomics, in situ sequencing, screenshot technologies at the European level with many collaborators as well. So you might see, for example, Kerstin Meyer, also from the Wellcome Sanger Institute, and many others as well. Besides data production, we also have data analysis, so also a full course on spatial omics technologies, integrated single cell, spatial transcriptomics, and in situ sequencing, and all of those lectures are now available even on YouTube. So Merry Christmas, that was the plan, but there is also a very collaborative effort, not only from Sweden, but also collaborating with Elixir Germany, and many other groups. So you can see Mats Nilsson, Joachim Lundberg, and Stanley Notion's lab, as well as Fabian Theis and Charité with Navid Ishak and Karolina Welby. And this year, Sweden has also now created the federated EGA, and we're also trying to help them to establish this secure way to deposit FASTQ data in Sweden, and this is also something that we are trying to get established. And that's it from my side. Thank you so much for the opportunity to be presenting here today. And this is a very collaborative work from many different groups. Most of them are already referenced in the slides. And if you have any questions, go ask them. They are in these posters. Please go there and ask questions tomorrow. Thank you.
Thank you very much. Okay, maybe one question, and then the rest of the questions we've been instructed to ask these people.
Thank you. I have a question on the eggplant, since it don't have a poster. So I wonder whether it can map samples between stage, so that then you can see the temporal progress on spatial data.
Yes, in theory, yes. So for eggplant, we try to map not only gene expression from spatial transcriptomics, but also, for example, in situ sequencing, MALDI-TOF, and image analysis, so for example, screenshot, all these different omics into the same reference. So it is possible to even also map different developmental times. It could be also, especially in development, because the morphology changes quite a lot, right? So it might make sense to map them at the different time points separately than at the whole thing. So yeah, it is possible, but it might not make much sense to combine everything into the same reference, because the reference changes in that case. All right.
Let's thank Paulo once again. Thank you. Okay, our next lightning talk speaker is Heather Etchevers from INSERM in Marseille.
No. Can you hear me? We're good? Okay, maybe a little higher. Sorry. It's further away that way, but okay. That works, great, thank you. So that's supposed to introduce my talk. Welcome. Could you load my talk, please? Anyway, great, here we are, thank you. I'm here to tell you today about some independent efforts to refine the human heart atlas during early development, and it's very encouraging to see the community addressing these issues of making our atlases more accurate, more useful, enriched with different modes of analyses. So I don't have to spend a lot of time saying, but different cell types in the heart, they are going to differentiate and appear at different times during development. And we know a lot about this from using animal models, but there are a lot of human-specific aspects to it, and probably cell types that are not represented here that we still have to discover in the human context. Relative to congenital heart defects, these are among the most common birth defects in humans. And if we're going to be studying what's going on very early in development, oh, how interesting, they change over time. We need to focus on these slightly later stages than had appeared when we first started this work as far as the human heart atlas. And so we decided to do a much less high throughput approach and a more focused in-depth approach on specific heart samples from these human embryonic and early fetal stages. And I guess I should point that way. There we go. And so to make a quick summary, because we don't have a lot of time, we did single nucleus RNA-seq on 3 hearts of 2 different sexes, so it's not a lot per individual, but we did do a lot of cells. And the reason for that is that we had found that in the data that appeared around the time of the pandemic, we were missing cell types that we expected to see in that data. And so we focused on these 2 stages, and then we also completed the data with some spatial transcriptomic approach using the 10x Visium platform at the time, and that gives you sort of the hard data about it. Just one caveat about that is that we tend to say that the spots that are present on these slides will cover a certain number of cells, and of course that's an average, and we already heard that averages are not ideal for portraying what's actually going on. So it can be more or less useful, but we found that crossing the data was very helpful. And so in our single nucleus RNA sequencing, we used prior knowledge to sort of manually annotate and try to figure out what clusters we were going to call what names. And with the spatial transcriptomics data, you can sort of take the spots. As I learned today, you can also take organoids and use them. It doesn't really matter what you use, to also cluster and come up with information that then you cross. And so the first question we wanted to know is that where are the cell types that we've isolated, and especially in this information, especially the very minority cell types. And I have to praise the phrase that rare cells matter. I totally believe that, and particularly for a developmental biologist, rare cells induce other cells to become what they could become. And so this is just one example of the kinds of things that we could discover by looking deeper in depth. So there were in fact not 1, but 4 different fibroblast subtypes that we could identify in those 2 hearts alone at those 2 specific stages. So we expect there's probably some greater diversity out there. And one of the cell types was spatially very restricted, and this is corresponding to valve interstitial cells. The reason there are 2 photographs here, one corresponds to mapping all of the spatial transcriptomics clusters back onto the sections, and the other corresponds to mapping all of the single nucleus clusters back onto the sections. And so they were quite concordant, and that made us happy. That was not true for all the groups. And then, of course, by looking at the most greatly differentially expressed cells, and we focus on this gene that had not been known to be important in valvular interstitial cells, we saw quite regionalized expression, which is encouraging to continue on with some functional work. I'm going to make a long story short, but thanks to a bioarchive deposit, we were able to connect with the laboratory at Memorial Sloan Kettering, where Yiqi Xu, I hope I didn't massacre your name, who's in the audience, presumably. We have not yet met, but we were able to collaborate on integrating our atlases from the single nucleus data. And they had done a very comprehensive atlas of earlier stages. And we said, let's extract the heart data and see what we can learn about heart development over time by also combining the data with what we saw from the earlier talk from the group in Sweden, that they also are refining. So we're going to be reiteratively integrating our atlases over time and over the years ahead, and that's a good thing for all of us. And so we saw that there was a lot of, how should I say, not redundancy, but a lot of synergy, that we could really help one another out. So this is going to help us to understand where, when, and what the cells are in the tissues directly. And so without going really into the data, which you'll be able to see in one of the posters, we are also using these three-dimensional reconstructions of sections using protein markers in the heart tissues in order to go back to the spatial transcriptomics and position those in space. Because, of course, you know what you've sampled, but you don't know everything else that is out of plane. And use a common coordinate system that's going to be based on peripheral nerves and blood vessels that are everywhere. And so for us, that enables a three-dimensional space on which we're then going to be able to map time and morphology. And so this was just some examples of markers that we then looked at in immunofluorescence over here that were positioned, but again, are new markers for these different cell types. So with that, I'm going to just stop here, I think, if I can get it to advance. Yes. And say thank you to all the team, and in particular to Stéphane Zaffran, who is also here. And he's going to present tomorrow in the second poster session, 22, if you want more technical details about what we've been doing and the questions we're asking. So all of this is not published yet. We don't even have the bioarchive up, but it'll happen soon, I hope. And thank you, of course, to all the other people locally who've participated in this, as well as our HUDECA collaborators here in France. This is the French consortium that I presented to you back in Tokyo a few years ago. And of course, our collaborators both in the US and China, and also on site for all the imaging information. So our website for UDECA is here. It's kind of newish, so be patient. And thank you for your attention.
Thank you. Thanks. Any questions for Heather?
Hi. Really wonderful work. I was wondering, when you were focusing on identifying rare cell types by sequencing lots of cells, did you find any small proportions, like cells that were coming up in small proportions across your different samples that you think are rare cell types that we've missed?
Absolutely. And it also helped us confirm some things that looked like they could have been noise. It was a little hard to define in some of the other data sets. So that's where I was saying we were really helpful in completing one another. Both the early embryonic data highlighted certain progenitor cell types that we saw more of in the later cell types, and we didn't really know what they were. And other cell types disappeared, such as second heart field cells. The neural crest cell lineages, there are two subpopulations that had been brought up in the 2019 paper, but their identity was a little bit, because there weren't enough of them. Now we have a much better handle on what they are and where they've come from. And crossing that to some of the mouse data has enabled us to make some hypotheses about where to go from here relative to those cells.
So, beautiful talk, Heather. You mentioned the cardiac conduction system, and I mean, were you able to see that in the light sheet?
We didn't stain for specific markers for it, but we could, and I think that's probably a next step, because I think we're very interested in looking at networked cells, in building that kind of scaffolding on which we can then, impose the rest. I think that it'll be very helpful for us to say, okay, what's the data we have now? Where is it? And what else needs to be done specifically for, I don't know, the atrioventricular node, or for very specifically regionalized structures, the valves as well. Okay, so with that, thank you very much.
Great. Thank you very much. So our next speaker is Dorit Hockman from University of Cape Town, and we're just getting set up.
Which is the pointer?
And that's the pointer.
Oh, is that the pointer? Okay, great. Cool. Thank you very much for this opportunity to show you what we've been doing in our lab at the University of Cape Town to explore gene expression and gene regulation in the maturing human brain. So I'm sure you're all very aware that we actually have made a lot of advancements in understanding the molecular composition of the adult brain. Through efforts of the Human Cell Atlas and the Brain Initiative Cell Census Network, we already have a detailed draft census of many of the cell types in the adult brain, and it's recently been extended to various regions of the adult brain. And so this work combines single nucleus RNA sequencing and various epigenetic factors to define the cell types in the adult brain. But what we don't know really is how do these profiles change over the course of brain maturation. And at the University of Cape Town, we've got an amazing opportunity to start answering this question. So we've, through collaborations with neurosurgeons at the local hospitals, we are starting to put together, well, we're putting together a biobank of tissue samples collected. So these are the access tissue samples that are collected during neurosurgery to treat epilepsy. And importantly, this is also unique in that these are data sets that are coming from people of African ancestry. And our biobank now spans ages from around 10 months all the way to adult. And we're using this now to build a locally relevant single-cell database, which we can now use to understand gene expression in the pediatric brain in comparison to adults. And to do this, we're using many of the approaches we've heard about already, so single-nucleus RNA-seq, spatial transcriptomics, and also bulk attack-seq at the moment in order to understand what's going on in brain maturation. So what I'm showing you here is some of our data, which includes 8 new data sets, which are shown here in bold, which include 5 pediatric samples, all from the temporal cortex. And to analyze them, we're integrating them with publicly available data sets from the temporal cortex. We have analyzed these data and used the label transfer approach from using the current Allen Brain Map medial temporal gyrus dataset to annotate our data, which has allowed us to identify 54 cell subtypes within this integrated dataset. And reassuringly, when we look at the markers that are being expressed in all of these cell subtypes, we can identify common markers for inhibitory neurons, our excitatory neurons, and our non-neuronal cells. And we're now doing a lot more work with this data set. So one of the aspects we've been looking at is to see if we can actually we can also see sample-specific minimal markers for these cell subtypes in our data to see if we can find markers that change over the course of brain maturation. And for more information on all of this work, please visit Christina Stein's poster, my student who's here, and she's actually now poster number 15 rather than 19 in this evening session. But obviously what we also want to know is what are the spatial dynamics of these cell types over the course of brain maturation. And so we've used Visium spatial transcriptomics to try and answer this question. And so what I'm showing you here is our data for 3 different age samples. And what we've done is use the cell-to-location tool to take our annotations from the single-nucleus RNA-seq data and to transfer them onto our tissue sections for the temporal cortex. And what's great is that we can see estimated cell abundances in the right places for these astrocytes and excitatory neurons in the top row, and as well for oligodendrocytes and inhibitory neurons in the bottom row. You can start seeing the characterized layered structure of the cortex. And what we also want to do is understand how these spatial profiles are changing over the course of brain maturation. And again, we're using cell-to-location's NMF approach to identify cellular compartments or co-occurring cell types, and to understand how maybe their distribution is changing with age. So for example, this cluster consists of a subset of inhibitory
cell types, which looked as if from like our initial eyeing of the data, look as if they may be more spatially confined in our adult section compared to the pediatric section where the cellular compartment seems to be more dispersed. And for more on this work, please visit another one of my students is here, Ravimbo Mishi. She's now poster 13, not 15, in this evening session. And the next step for this is obviously to understand how gene expression profiles associated with the cell clusters are changing over the course of brain maturation, and do the profiles vary with age. So I'll show you what we're doing so far with our single nucleus RNA-seq data to answer this question. We've taken many approaches, but I'm showing you two of the approaches here to do differential expression analysis. So firstly, we've using a pseudobulk approach where we're getting pseudobulk values for each of our cell types for each sample. We've grouped the samples into epochs, as highlighted in these color codes here, and then used DESeq2 to identify differentially expressed genes. We've also done a supervised pseudotime analysis using quite a new tool that's recently come out to take advantage of all the nuclei rather than using a pseudobulk approach. And what I'm showing you here is just a very short snapshot of the data for this particular neuronal subtype, shown in the UMAP over here. And this list of genes is 16 genes that were identified by both of these approaches, so we're quite confident in them. And you can see from this dot plot that it includes genes that are both increasing over the course of brain maturation, as well as those that are decreasing. And reassuringly, when we look at what we know about some of these genes, some of the genes are known to play a role in brain maturation. So for example, this FNBP1L is a gene that was found in our DESeq2 analysis to be in a group that decreases in expression level over time. And this is known to play a role in actin cytoskeleton assembly and neurite outgrowth, which are both incredibly important for neuronal maturation. They've also been linked, this gene's been linked to intelligence in adult and children, as well as being associated with neurodegenerative disorders like Alzheimer's disease. So it's a very, I think it's a very promising result. And again, sorry, before I move, sorry, going in the wrong direction. Please do visit Christina's poster for more information on this. So finally, as I said, we also have bulk attack-seq data. And this data is to help us try and find the regulatory elements that may be driving these gene expression changes over time. And so once again, this is the FNBP1L locus showing our bulk attack-seq data sets from a variety of different ages, alongside some single nucleus attack-seq data that's publicly available. And we're hoping to generate our own single nucleus attack-seq. We are at the moment doing that. But the idea is to compare our bulk attack-seq to single nucleus attack-seq and try and identify cell type-specific enhancers. And then finally, once we find these enhancers, we also want to be able to test whether they actually indeed do drive cell type-specific gene expression. And so we're testing in vitro enhancer reporter systems in our lab using human organotypic brain slices, so from these same patients that go into our biobank. So we're using an electroporation approach to deliver these constructs into the brain slices. And this is just one of our positive controls over here, where we've delivered this ubiquitously expressed construct into one of these slices to beautifully label these cells. And for more on this work, please also visit Justine van Grenen's poster on tonight, and she's now poster number 16. So yes, in summary, I've shown you what we're using this unique tissue resource to describe the cell type-specific gene expression changes that may be occurring during human brain maturation. And this is already starting to form an important locally relevant control for understanding how the cells, these gene expression change, these cell type-specific gene expression profiles are altered in response to disease, in particular locally relevant diseases like TB meningitis. So finally, before I stop, we have a postdoc position available in my lab. So if you're interested in coming to work at the beautiful new Neuroscience Institute under Table Mountain, please do get in touch. And then thank you to all my incredibly hardworking and excellent students and my collaborators and funders. Thank you.
Thank you. Any questions?
Yeah, Yeah, wonderful, wonderful work. And we rarely see these postnatal kind of studies because those are from pediatric cases to adulthood, right? I was wondering if I understood correctly, you have taken chunks of the frontotemporal lobe, right?
So this is all temporal cortex tissue that I've been showing you.
Have you compared these with biopsies from patients with frontotemporal dementia? I think we might have a biobank in at least one, perhaps two in France with these kind of biopsies from postmortem brains that are actually exactly the same regions that have been taken, and comparing your data set with eventually generating a new data set from frontotemporal dementia.
No, we haven't done. So you're saying compared to the available post-mortem tissue to see if we see something similar in ante-mortem samples, or
to see in the pathology of frontotemporal dementia will particularly affect those areas that you've been dissecting.
Yeah.
So you will have a signature of a reference atlas that you've been building compared to divergence in a pathology that affects exactly the same anatomical structure.
Yeah, that's a very good idea. No, we haven't done that. I think we've focused mostly on integrating with other similar data sets, like that are also collected during surgery, just to try and remove as many artifacts as possible. But yeah, that would be a really powerful analysis to do as well. Yes.
We have time for another question. Yeah, okay.
Thank you very much. That was very exciting. Very quick question about the samples that you've got. Are they all a similar type of course to have a post-mortem, or I guess I just wondered what the metadata was that was available.
Yeah, so we have, these are all from living people who've had surgery to treat epilepsy, and so we have the full patient records available through the neurosurgeons. I don't physically have it myself, but if we could actually get that information to include in the metadata. Like I don't know any of the information, but we do have it from their patient records.
Epilepsy arising from the temporal cortex, and the surgery would be to ablate the kind of like foci triggering the kind of seizures.
Exactly. So they have to go through. It's usually deeper, and have to go through the temporal cortex in order to get to the tissue. So this is the access tissue to get to the focus. So the epileptic focus has gone off to the pathologist, and maybe we can get access to that information as well from the actual epileptic focus region. But we're treating this access tissue as normal. But obviously we also have access to all the medical data. So if we see something coming up that's like, oh, actually this is epilepsy, then we can find that in the data as well. So that's always in the back of our head, is that this is as normal brain tissue as you can get access to in terms of living brain tissue, but it's going to have the caveat of coming from epileptic patient.
Okay. Thank you very much to all the lightning speakers and in this session. So I think the next session is a poster session and reception. Okay, so are there any other announcements to make before we go? No? Okay, thank you.